Our goal was to understand why the photoreceptor transcription factor CRX activates transcription at some of its target sites and represses transcription at others. Our analysis of genomic sequences suggested the hypothesis that the number and affinity of CRX binding sites in a regulatory sequence determined whether CRX would activate or repress from that sequence. Specifically, our analysis suggested that sequences with a high total affinity for CRX, that is, with more binding sites or higher affinity binding sites, would drive transcriptional repression, while sequences with a moderate affinity for CRX would drive activation instead. To test this hypothesis, we constructed a set of synthetic regulatory sequences that contained only CRX binding sites. Unlike genomic sequences which might have undetected sites for other transcription factors, we have complete control over the design of synthetic sequences. Therefore, by varying the number and affinity of CRX sites in synthetic sequences, we could directly test our hypothesis that transcriptional activation and repression are encoded in a regulatory DNA sequence by the affinity of that sequence for CRX. We constructed a reporter gene library of synthetic sequences that consisted only of different combinations of high, medium, or low affinity CRX sites. We also designed a set of sequences in which one CRX site was replaced by a site for NRL, another photoreceptor transcription factor that is known to interact synergistically with CRX. We tested the activity of these reporter genes using our massively parallel reporter assay in newborn mouse retina. The results support our hypothesis that activation and repression can be encoded in a regulatory sequence using only CRX binding sites. The x-axis shows the total CRX affinity of the sequences, while the y-axis shows reporter gene expression. The dashed line indicates the basal activity of the rhodopsin promoter. Lower affinity sequences activate reporter gene transcription above basal levels in our experiments, but as total CRX affinity is increased, sequences are more likely to repress below basal levels. This shows that CRX binding sites alone are sufficient to encode transcriptional activation and repression. An NRL site overrides repression. When we add an NRL site to sequences with high total CRX affinity, instead of repression, we observe transcriptional activation. These experiments demonstrate a remarkably simple way of achieving both activation and repression in a single cell type using binding sites for only two transcription factors.